Good morning, good afternoon, or good evening to all, depending on where you're joining us from. On behalf of the National Society of Biotechnology and Engineering, SONABIN, and the organizing committee of the International Congress of Biotechnology, Engineering, and STEM 2025, I warmly welcome you to today's session. We are truly honored to have such a diverse and engaged international audience joining us via Microsoft Teams. This Congress brings together researchers, professionals, students, and entrepreneurs from across the globe to exchange cutting-edge knowledge, innovative practices, and technological advancements in biotechnology, engineering, and STEM fields. Thank you all for your active participation and enthusiasm. It is my great pleasure to introduce today's distinguished speaker, Magister Martin Stacho. Dr. Stacho began his academic journey at Charles University in Prague, where he studied the invasiveness of metastatic sarcoma cells under the supervision of Professor Jan Brabec, an experience that sparked his passion for research. He then pursued his PhD at the Max Planck Institute in Dresden, focusing on the role of photosomes in bone-resorbing osteoclasts. His work involved identifying regulatory proteins and uncovering the function of phosphoinositide pathways in photosome dynamics using mass spectrometry. His postdoctoral research at the Max Perutz laboratories in Vienna delved into the physiological processes of autophagy, where he applied CRISPR-Cas9 and microscopy techniques to examine how phosphoinositides orchestrate the condensation of protein cargo through phase separation. In 2017, he joined the Institute of Molecular Genetics in Prague, working with Professor Pavel Hosak to explore the role of phosphoinositides in nuclear architecture, particularly their function in nuclear subcompartmentalization and protein localization. Currently, Dr. Stacho serves as a junior group leader at the Institute of Biochemistry and Experimental Oncology at the First Medical Faculty of Charles University. His research continues to focus on nuclear organization, gene expression regulation, and cancer progression. Please join me in welcoming Dr. Martin Stacho.
Hello, thank you very much for kind invitation and introduction. I hope everyone can hear me well. I will now
Yes, we can hear
share.
you well.
Fantastic. So let me share my screen. Uh, uh, uh. So you see my presentation, I hope, right? So it's all good. So thank you very much for kind invitation to this meeting. Today I wanna talk about the spatial temporal organization of super enhancer structure in cancer cells. So before we start, I will provide a very brief introduction. What is the organization of life matter in the cells? So I will talk about the nuclear architecture. If nuclear architecture, you can imagine as the subcompartmentalization of the life matter, which ensures that various biological processes happens in distinct areas, at the areas where they should happen. And in the cytosol, usually it's ensured
Excuse me,
by,
Dr. Stajo, would you
yes,
please share your presentation in the presentation mode?
sorry, is it
Yes,
better
there
now?
you go.
Good.
Thank you.
All right, sorry. Thanks for letting me know. All right, so we have in the cytosol subcompartments, or sometimes called organelles, which are typically delimited by membranes. However, when we look at the cell nucleus, the situation is a bit different. We see that the only membranous structure is the nuclear envelope, whereas inside we don't have any membranes. However, these regions are also functionally or structurally distinct from their environment, And we can talk about nuclear bodies such as nuclear speckles, where all the splicing machinery is stored, PML bodies, Cajal bodies, or even nucleoli as a typical example of nuclear structure. And all these structures share a common feature. They are, how do I, okay, sorry. Yes. So they are all formed by a process which is called phase separation. Phase separation you can imagine as a glass of water where you pour oil. So these are two liquids which have a different physical-chemical properties, and it tends to form kind of liquid-like droplets. Similar situation is also in the cell. They are certain molecules, especially proteins, which have special structural features enabling them to form these liquid-like droplets. And this is usually happening through the local increase of the concentration. So if you look at this cell nucleus, this is the same cell just captured at the different time points, and you see that from this, let's say, granule pattern, we eventually go to the situation when the cell nucleus is completely filled with liquid-like droplets. This is happening due to an increasing concentration of the constituents. So when the concentration of constituents increases, we have the formation of so-called condensates or bodies, which eventually merges in order to form bigger ones. So as I said, it's a function of concentration. So on this axis we have a increasing concentration, and on the y-axis we have parameters like temperature, ion strength, pH, etc. So basically, when we are at the beginning, we have a low concentration of the constituents, and eventually we enter this two-phase regime, which is representing the two-phase behavior uh, of the condensates. So, but what is it good for? So we can imagine that the condensates, the accretions of the life material within the cell, it concentrates locally the constituents of the biochemical reactions, but as well, it produces the surface at the interface between the actual condensate and the neighboring environment, and this surface might serve as a kind of catalyst, because if the reaction happens adherent on on some surface, the effective interaction of the molecules is much more likely than if it would be in insoluble environment. That leads to easily overcome the energetical barrier of the biochemical interactions. So basically, this fulfills the requirements to be called the catalyst, and this is exactly how the condensates work in the cell. Why the condensates are formed via the phase separation, the absolutely crucial aspect is the possibility of multivalent interactions. So here we have two scenarios. One is the protein with let's say globular domains interconnected with the linkers. They are very flexible, but these domains, they enable the interaction with their partners. Uh, and a similar situation is with so-called intrinsically disordered proteins. So these are proteins which typically don't form these globular functional domains, but they rather have or possess the stretches of typically uh, charged amino acids. And these charged amino acids, they kind of enable the multivalent interactions among the different molecules. So here you can imagine it as this as it is depicted on this picture. So we have basically a bundle of the molecules with the stickers, the domains or stretches which are able to form the intermolecular interactions, and eventually without no addition of external energy, it leads to the formation of droplet or the condensate. Now, the net charge and the hydrophobicity of the constituent is absolutely crucial parameter for the localization of molecules within the cell. So here I took this from one paper published in 2022. What the authors did, they designed artificial polypeptides, and these polypeptides, they were composed from stretches of arginine or lysine amino acids. And depending on the number of these amino acids, they dictate basically the charge. You know that these amino acids, they have the very same charge. However, they have a slightly different physical chemical properties, and increasing in this scale basically on the left side, the peptides can be targeted into a different regions of the cell nucleus. So you see that here when the net charge is plus 0.2, this containing arginine goes into nuclear speckles, whereas the arginine containing ends up in the nucleoli. So this is very important when we look at numerous transcription factors. So these are key regulators of gene expression machinery. Their C termini is often very acidic, and this is quite well conserved across the different species, and people just lightly figure out that this is quite important feature. And if there is a gain of function frameshift mutation, so this is just one nucleotide, let's say deletion or addition, it leads to the frameshift, which changes this very negatively charged C tail into a very basic or positively charged termini of the mutated proteins, of the mutated transcription factors. And what happens in the cells? So always in this, let's say, first row, we have a wild type situation, whereas the lower, this is a pathological mutation which was identified in certain patients, and it leads to the formation of the so-called KR-rich motifs. And you see that in these at least six examples, they are much more, but I just wanted to show these six, that basically the mutation, the frame shift, leads to a huge difference in the localization of the transcription factor. So they tend to relocalize in these phase separated condensates, suggesting that such a minor change in the code leads to a very dramatic changes in the protein localization to completely different subcompartments of the cell nucleus. And don't forget that we are talking about transcription factors which are tightly regulated in order to define the expression patterns of the cells. These changes, they often happen also in development of cancer or the cancer progression. And it's really important to mention here that studying the condensates, the biomolecular condensates, is also important because in future they might serve as or for the targeted a treatment. So here we have in vitro prepared condensates from different factors like mediator complex, subunit, BRD4 as the co-activator of transcription, etc., etc. And these proteins, they do phase separate. And if these authors incubate those condensates in vitro with fluorescently labeled drugs like anti-cancer drug cisplatin, JQ1, that's the BRD4 inhibitor, or mitoxantron, what is quite novel anti-cancer drug or DNA intercalator, you see certain type of specificity for distinct types of condensates. And this specificity might be used in future for exact design of the chemical compounds which would target specific condensate, let's say only in the cancer cells, with no harm to healthy cells. Now, I talked briefly about charge, but here this is very important to mention that in case of this thing condensate, we need that the charge is in the balance, in the kind of meta state, uh, and this is ensured in or by help of RNA very often. And when we are here, basically in this metastable state, we have a condensate. But if we move from the balance to either left or right, the condensate dissolves. And as an illustration, I add here very simple experiment where I overexpress CLK, that's the kinase which phosphorylates SR proteins. So these proteins are taking part in the formation of certain condensates in the cell nucleus. And you see that the wild type CLK expression leads to disassembly of these condensates. Okay, so when we have the overlay of the signals, we see that only the cells which overexpress the active kinase dissolves the pattern. And it's really due to a enzymatic action, due to the kinase function of the protein, because if I used kinase that mutation, the overexpression of the enzyme does not lead to disassembly of this condensate. This post-translational modification, as already described, phosphorylation changes the meta state of these condensates, and it leads to increase the negative charge, eventually dissolving the condensate. So now, in this respect, I would like to talk about phosphoinositides, since these are the molecules which are bearing negative charge. So you know that these are basically lipids, which are typically embedded in the membranes. So we have inositol ring, which can be phosphorylated at the various places, leading to the formation of 7 different types of the phosphoinositide. So this is kind of a code which is recognized by enzymes with specific recognition domains. And the best studied example of the phosphoinositide code is basically in the vesicular transport, or so-called trafficking in the cells. So you see on the legend that there are different, let's say, subpopulations of distinct phosphoinositides, and they participate in the directed movement of vesicles, and they attract different types of proteins. And this is quite complex phenomenon. But if you look closer, the nuclear environment in this scheme is blank. It's empty. However, the nuclear environment also do contain phosphoinositides. It's still mystery how they are localized there, because they have this very hydrophobic acyl groups, which are typically embedded in the membranes. However, if we look in the nuclear environment, we see for this phosphatidylinositol 4,5 bisphosphate that it localizes to a different areas. So this is a super resolution microscopy showing the indirect immunofluorescence, and this is the electron microscopy, and we can distinguish 3 pools. So first is the pool which localizes to nucleoli. Then we have, so here it's in blue. Then we have in yellow, these are localized to nuclear speckles. These are these very bright, quite big condensates. And then we have a dispersed pool in the nucleoplasm. And if we look closer, especially by the electron microscopy, here this is immunogold labeling, we see that these are around 100 nanometers rounded structures, which are actually dispersed in the cytosol, nucleoplasm, sorry. And if we do a co-staining, immunofluorescence co-staining, we see that they often localize in the proximity to transcriptional factors and polymerase 2. Therefore, there was an idea that these structures might have something to do with the regulation or assembly of polymerase to machinery. Therefore, the PLC enzyme used to cleave the PIP2, the phosphatidylinositol, leads to inability of the cells to incorporate fluorescently labeled RNA, suggesting that really the lipid is necessary for the proper transcription in the cell. And then the question was, what are the nuclear proteins localizing or interacting with this nuclear phosphatidylinositol phosphate? Therefore, I designed um, mass spectrometry based approach. I don't want to go into details too much, but the experimental setup enabled to identify the exact regions of the interaction with the molecule. We based this on limited proteolysis attempt, and the mass spec provided us well, let's say almost 200 proteins, and we could divide them depending on their enrichment in the analysis on so-called exposed and hidden interactors, where we propose that the exposed are the direct interactors, whereas the hiddens are indirect, so they need another protein or other molecule in order to interact with the PIP2 molecule. The further analysis, this was gene ontology analysis. That's basically one a bioinformatical approach enabled or pointed out that most of these proteins are connected to polymerase 2 transcription and the regulation uh, of the recognition of promoter. When we look at the cellular compartment, these proteins tend to localize. We indeed identify that they often localize to nuclear speckles, nucleoli, and as the algorithm calls it, non-membrane bound organelles. So these are the condensates. And I mentioned that this approach enabled the identification of the exact um, portion of the proteins which are in the contact with the lipid, and the analysis of these regions showed that they are quite hydrophilic, suggesting that these proteins do not interact with the acyl groups because they are hydrophobic, whereas they preferentially interact with the head group, the inositol group. That means that these proteins indeed recognize the code of the phosphates. I already mentioned the importance of RNA in the formation of condensates, but here I would like to emphasize that the RNA is so super important in the formation of nuclear architecture. All these regions or functional bodies are assembled due to RNA, which serves as a kind of bridging factor, which brings the constituents together. Um, Therefore, we wanted to know whether there are molecules which would interact directly with the phosphatidylinositol phosphate. So this study, I only participated. This is collaborative effort with Professor Castaño, and he got an idea how to attempt this problem. So it was basically using phosphatidyl inositol coated beads, and these beads were incubated with isolated RNA, and eventually the sequencing provided us the data and sequencing of and sequences of the RNAs which directly interact with this lipid. So the bioinformatic analysis provided us a list of the RNAs. However, we picked basically two top hits. One I called AU1 and the second AU2. The reason is that these RNAs are basically enriched for these nucleotides, so A and U, and these are consensual sequences. So it doesn't mean that this is a particular sequence existing in the RNA, but this is a consensual sequence. So it might be present in numerous RNAs in the cell. And we try to do fluorescent in situ hybridization of the cell nuclei. We use these RNAs fluorescently labeled, and you see that indeed they show pattern which is um, kind of overlapping with the PIP2 signal. So here this is in higher resolution in the zoom. And this one, this AU2, it showed only one specific region, which is also overlapping with the PIP2 signal, but it shows different localization, suggesting that they are different specific RNA sequences, which would be in interaction with distinct pools of nuclear PIP2. So we wanted to know a bit more about this. So we picked AU2. That's the one which shows the let's say this kind of a spot appearance, and then we tried to search for the let's say interactors or the proteins factors which would colocalize with this structure, and we presented here few like MYO1C, mediator 1, BRD4. So these are all transcriptional factors, quite important, and actually constituents of so-called super enhancer condensate. We also showed that they do colocalize with the 53BP1 protein, and we confirmed that one of the RNA, which was lately identified as the RNA which contains this consensual sequence, localizes to the same spot. Okay, so we did fluorescent in situ hybridization, and we identified that this HI and R RNA localizes to these factors, and moreover, it localizes to HNRNP1, A1, and that's the marker for so-called perinuclear compartment. And this is the compartment which was in 2001, if I'm not wrong, discovered by Professor Spector as the compartment which is formed exclusively in cancerous cells, and it has something to do with the aggressivity of the cancer of the cancer cells. However, the exact function is still unknown. But our consensual RNA, which is interacting with nuclear PIP2, is there from whatever reason. So we wanted to know what are the proteins which do interact with nuclear PIP2 in presence of RNA, and we used a trick. So here this is the cell nucleus where we localize PIP2, and we treat these cells, semi-permeabilized cells, with RNase 3. This RNase basically dissolves or cleaves the RNA, which we wanted, and you see that we reduce the signal of PIP2 in the cell nucleus. This signal decrease is specific in nucleoplasm and also in the nuclear speckles, and it allowed us to design mass spectrometry-based approach where we basically use this knowledge to identify the proteins which binds to PIP2 only in the presence of RNA. And here we have 168 proteins which do so. And these proteins we call RNA-dependent PIP2 associated nuclear proteome, or RDPA in short. We analyzed what these proteins are. So these RDPA proteome is enriched for proteins which do bind RNA. They can phase separate, and they can phase separate due to the presence of intrinsically disordered region. So this is the region I was talking during my introduction, um, where they have these charged amino acid stretches for the multivalent interactions. Moreover, these stretches belong to one of this family of motifs, of these Kr-rich motifs. So you might again remember that these Kr-rich motifs, these arginine, lysine arginine-rich motifs, are important for relocalization of transcription factors in cancer. So then we were analyzing a bit further in detail these proteins. We figure out that their intrinsically disordered regions are bimodal, so they have a part which is acidic, then the part which is basic, and there is a valley which kind of separates them pretty well. We figure out that their intrinsically disordered regions are significantly longer than in other data sets, than other proteins. We found out that they are a bit more basic because they do contain these Kr-rich motifs, and that they are hydrophilic a bit more than the other data sets, suggesting that indeed they are in the, or they might be in the contact with hydrophilic charged uh, phosphorylated inositol of the PIP2 molecule. So here we use a different gene ontology approach where we basically analyze the list of identified RDPA proteins and figure out that they are involved in regulatory processes around the RNA pol transcription, processing of RNA, and so on. So we took also the reverse attempt. So we took the motif, the Kr-rich motif, and apply so-called SLIMS analysis. So basically, you can imagine it very simplified that you would blast basically the proteins, all the let's say genome, and identify the proteins where these motifs would be enriched, and then you would do geoanalysis. And then you have a cellular compartment, molecular function, and biological process these proteins participates in. So again, they do localize to nuclear speckles, spliceosomes, nucleoli, etc. Their main molecular function is the RNA binding. And the biological process is the RNA processing, splicing, ribosome biogenesis. So all the processes which happens at the nuclear compartments like nucleoli um, and nuclear speckles. So all this fits surprisingly well, and that we wanted to see how it looks in real. So we picked few. I will just show you one. That's BRD4. That's a notoriously known a transcription coactivator, which forms condensates via phase separation. So in reality, it looks like these dots. So these brighter spots. These are condensates. And when we localize them together with PIP2, with the lipid, we see that these condensates are often in the proximity or at the surface of the PIP2 signal. So we did the analysis, and we figure out that indeed they do co-cluster in non-random manner. Now the question is, what would happen with these BRD4 condensates if we modulate PIP2 level? Because we believe that PIP2 as the recognition wedge, let's say, would serve as the condensation center. So we tried to diminish the level of nuclear PIP2, and we use depletion of this kinase. So this is the kinase which leads to the formation of this PIP2 in the nucleus, and you see that in comparison to the wild type non-treated cells, we have less condensates. In contrary, when we deplete this phosphatase, this SHIP2, we get more of nuclear PIP2, and we also get more condensates. So after the quantification, we see a tendency where modulation of the PIP is actually correlated with the number of BRD4 condensates. This leads us to a kind of suggestion that indeed there is an intricate relationship between PIP2 level and BRD4 condensate. And we wanted to ensure that this is due to the effect on the PIP and not the depletion of the enzyme. So we use SHIP2 inhibitor, that's a commercially available inhibitor. You see that it potentiates the level or increase the level of nuclear PIP2 and also potentiates the or increase the level of nuclear BRD4 condensates. That's these data are in accordance with the previous observation. And now going back to the role of RNA. So if the RNA is attracted or bound to PIP2, which eventually leads to the formation of condensates, we assumed that depletion of the RNA must lead to inability of the protein to form condensate. And indeed, this is what we observe when we treat the cells with the RNAs. So we remove the RNA and we have basically reduction in the condensate formation. Again, this is in accordance with the hypothesis. That's fantastic. But these are still quite indirect because this is done in very complex environment, which is the cell. And we wanted to confirm these by the, let's say, minimal in vitro reconstitution system. So this is a very, I mean, it's not so trivial to do it, but the idea behind is very trivial. So we took polymerase itself. The polymerase is known to participate in the formation of the transcriptional condensate together with BRD4, and we have its C-terminal domain conjugated with GFP in order to track it under the microscope. And here we have three conditions. So first of all, you see that the polymerase 2 forms the condensate in vitro, and we then mixed it with very small liposomes mimicking PIP2. You remember that these PIP2 structures are around 100 nanometer size, and we prepared the liposomes of exactly that diameter, but they had a different composition. So either they had just phosphatidyl inositol, or they had phosphatidyl inositol 3,4, or our phosphatidyl inositol 4,5P2. And as you can see that only in this case, the phosphatidyl inositol 4,5 increases the size of Pol2 condensate, suggesting that this potentiates the formation of it in vitro. Now we need to kind of go back to the cells. So we took the polymerase marker. So that's the marker for active polymerase, which is phosphorylated at serine 5. That's the one which actually does the business. And we have a system where we can increase the amount of the polymerase condensate in nucleus. So we deplete this protein, and suddenly we have more of these condensates. Okay. And then we wanted to see whether the manipulation with the condensate number leads to changes in transcriptional pattern or transcription itself. So we again incubated these cells with 5-fluorouridine, which incorporates during the transcription into the nascent RNA. and then we can measure it by microscope. And you see that after the manipulation, we have significant reduction of the signal of the incorporation, suggesting that the transcription is diminished. And we wanted to confirm this by measurement by qPCR of certain or chosen housekeeping genes. And in basically all the cases, the manipulation, the increase of the nuclear pull to condensate leads to reduction of the transcription or the amount of mRNA for these housekeeping genes, suggesting that really the number of the condensate, transcriptional condensates matters, that it really impacts the transcription heavily. So here I would like to just sum up basically why this is important to study phase separation in cancer. They were already, or the amount of studies showing that liquid-liquid phase separation is connected to progression of cancer is increasing, and that the players are really proteins and RNAs. So formation of the aberrant subcompartments, you saw that I showed you this perinuclear cancer cell specific compartment is actually formed due to multivalent weak interactions. And the liquid-liquid phase separation increases the reaction kinetics, where it provides to the surface and accumulates the constituents of the reactions. The condensation centers trigger the formation. So you see that even at the physiological concentration where the protein should not normally condensate, but it does if you provide the condensation center. I showed you the case for this phosphatidylinositol 4,5. The formation of condensates, it buffers the fluctuations of the concentration. Therefore, it's helpful to maintain the reactions, basically the kinetics, even if the constituents are very low. And the condensates forms diffusion barriers. First of all, they make the interface between in and out, and for some of the molecules, this in is not accessible, basically, just based on the physical chemical properties of this environment. So we can control the condensation via charge, as I showed you. Therefore, post-translation modification or even drugs can be used to manipulate this. And the condensates, they enable the crosstalk, because imagine you have a condensate of transcription machinery and you have a condensate of pre-mRNA splicing, and these condensates can actually merge, or they get in contact, where the interface provides the or meets these two processes. Therefore, co-transcriptional splicing actually might happen, as we see for more than 80% of all the splicing happening in the cell. And that's kind of for the future. I believe that the targeted enrichment of anti-cancer drugs happens in the condensates, and I believe this can be manipulated by chemistry, by the synthesis, smart synthesis. So here, I would like to thank you for your attention. I would like to thank my collaborators like Pavel Hozák, Enrique Castaño, the members of our newly born team. I would like to thank the funders, and I'm happy to take your questions. Thank you.
Thank you very much, Dr. Estajo, for that insightful and inspiring presentation. Your work highlights the complexity and elegance of cellular processes, and we are grateful for the opportunity to learn from your experience. At this time, we would like to open the floor to questions. If you have any questions, please feel free to raise your hand or type it in the chat. We'll address as many as time allows. I can already see that some questions have been submitted in the chat. So if you'll allow me, I'll begin by reading them aloud. So
Yes,
the
please.
coordinador of posgrado is asking, the expression of CLK1 leads to a diffuse PIP2 pattern. Have you checked for the level of POL1 or POL2 transcription on those cells? Does that get reduced?
Yeah, thank you very much for this question. I have to admit we haven't tried yet to check this. So this was for us more like a proof of principle, whether the introduction of the charge actually changes the condensation capacity of the constituent. Um, but it's true that basically the next step would be to check whether it has an impact on the actual biological process. So one of the definitive and kind of a suspects to check would be, of course, the overexpression of CLK. Yeah, that's a absolutely valid comment. Thank you.
Thank you. Yahaira de Jesus is asking, good morning, doctor. Could you tell us how the spatiotemporal organization of superenhancer changes during tumor progression and how the nuclear localization of a superenhancer influences the expression of He oncogenes? And
Oh.
whether the types of cancer that exhibit significant reprogramming of superenhancers are those that regress in patients who previously had a type of cancer.
Yes, thank you very much. That's a great question. Um, Here, I have to admit, I'm a bit sorry because so this topic is still in its infancy. So we suggest that indeed, so I mean, all the cancer types are different, and patient by patient, basically, these mutations they do differ, and it's usually a collection of hundreds of the mutations when the cancer develops. So definitely there will be differences in the expression patterns and also in the formation of the condensates. However, to my knowledge, there are not yet that many data which would specify, let's say more directly what I mean. There are already some studies which say, okay, so these are the genes which are highly elevated during the progression of this and that cancer, but the knowledge is still very limited, and we are still at the very beginning. So now what we try to do is to understand the fundamentals of the structural changes, and then they are already some very initial data, but I think yeah, still really we are at the beginning. So I cannot provide more a specific answer.
Thank you. Jahair is also asking, Doctor, could the spatiotemporal disorganization of superenhancers be a viable therapeutic target in cancer treatment? Could computational simulation of superenhancer dynamics help predict phenotypic changes in cancer? Could the spatiotemporal dynamics of superenhancers be used as a diagnostic tumor biomarker?
Yes. So first, whether it can serve as a therapeutic target, I hope so, and I really believe so. The problem is um, we are at the moment very limited in tools in this field. So there are already tools which triggers the formation of the condensates. You can potentiate the local association and eventual phase separation of different proteins, including those participating in the formation of superenhancer. However, we still don't have tool which will specifically dissolve this and that condensate. This is something what this field really needs. And I mean, I have to say it specifically because in the cell you also have many physiologically relevant and helpful condensates. Okay, So if you would just dissolve all the condensates in the cells, then the cell wouldn't be happy. So we need something what is very specific. Unfortunately, it's still not available, but I believe so. We are also trying to come up with one system, but it's very challenging. Second part of the question, um, I worry I cannot answer this because I'm not really an expert in modeling. But I assume since this field is expanding and progressing quite heavily in very near future, I believe we can basically simulate and model basically whatever we want. So let's see. And the third part was whether it can be a diagnostic marker. Definitely, because I mean the super enhancers are typically formed during the embryo development. Okay, so basically cells they do form the super enhancer whenever they need a burst of expression of certain proteins. And here, unfortunately, the cancer cells use it for this purpose as well. So I do believe it can be used as a diagnostic marker.
Thank you. So Maria Concepcion is asking, PIP2 plays a key role in a signaling pathways, PI3K/AKT, involved in the development and progression of many cancers. Based on your knowledge, do you think it could be used as a cancer marker in the future?
Yes, not only this, but so here this is currently what we actually study in our laboratory. So when people study phosphoinositides, they usually study them in the cytosol. So this is the example what was just mentioned. So this is PI3 kinase, AKT signaling. This is I think in 80% of all the cancers, this is the mutated pathway. So this is absolutely crucial in cancerogenesis. So we need to understand what's going wrong there. That's the first thing. But the second, that people who understand or well trying to understand this pathway usually don't study what's happening in the cell nucleus. So there is all the machinery, all the metabolism for production of all the distinct phosphoinositides also in the cell nucleus. So we don't know. So if we affect the one in the cytosol, which regulates, let's say, migration of the cells or invasivity of the cancer cells, we don't know what happens with those phosphoinositides which do regulate transcription in the cell nucleus. So what we are trying now to kind of put it together and study it as a complex problem. So if we modulate the cytosolic pips, what happens with the metabolism in cell nucleus and vice versa. So I have to say we were quite successful recently in developing a tool which would attenuate only nuclear phosphatidylinositol 4,5 pool, leaving the cytosolic intact. And I think this tool might really provide us a power to distinguish between the functions in the cytosol and the nucleus. So I think this is more complex, actually, question which we are trying to address. Yeah,
Thank you.
thank you.
This question is from Luis Carlos Rodriguez. Is there evidence of the type of physical-chemical interactions that exist between phosphoinositides and nuclear proteins, and whether this could be altered by some pro-oncogenic proteins?
Um, so the nature of the interaction. So it's usually electrostatic. Okay, in most of the cases. So when we study this intrinsically disordered proteins, so I showed you that basically um, this interaction between negatively charged head group of phosphatidylinositol is interacting with this Kr-rich, so arginine, lysine, positively charged amino acids. However, as I showed you, there is also RNA in this respect. So in principle, the logics or the common sense shouldn't allow two negatively charged molecules to interact. but they are three molecules. So the lipid negatively charged, then negatively charged RNA, and positively charged protein. And all this somehow, we still don't know how, but bundles together and form the condensate. How to um, how to actually dissolve it? I mean, you can dissolve it, for example, by salt, very high salt concentration. But the concentrations are that high that basically the cells would lyse. Okay, so uh, the question is how to approach it. There is also a drug which was like some years ago commonly used to dissolve, forgive me, to dissolve phase-separated condensates. It's called, it's alcohol. It's 1,6-hexanediol. So it does the same business, but the problem is that it has so huge side effects that you basically cannot use it in the cell. So basically, in a sense of few hours, you kill the cell. You dissolve the condensate, but at the same time you kill the cell because it's non-specific. That's why I said we need something more sophisticated and more focused, more targeted. That is specific for this particular condensate. Yeah. Thank you.
Thank you once again to everyone who joined us and actively participated in today's Q&A session. On behalf of the National Society of Biotechnology and Engineering, SANABIN, and the organizing committee of the International Congress of Biotechnology, Engineering, and STEM 2025, we would like to express our sincere appreciation to Dr. Martin Estajo for his inspiring and thought-provoking presentation. As a token of our gratitude, a certificate of recognition has been prepared in honor of Dr. Estajo's valuable contribution as a speaker at this international congress. We will now share the certificate on a screen, and I will take a moment to read it aloud. The National Society of Biotechnology and Engineering grants this constancia to Dr. Martin Estajo for his valuable participation as a speaker at the International Congress of Biotechnology, Engineering, and STEM, presenting the keynote address entitled Spatial Temporal Organization of the Superenhancer Structure in Cancer Cells. Once again, thank you, Dr. Stajo, for your generosity in sharing your time and knowledge with us. This concludes our session. We invite all participants to continue enjoying the rest of the Congress and to stay connected throughout the day. Have a great day or evening, wherever you may be in the world. Thank you very much.
Thank you very much. Thank you.
Thank you.
En 2 minutitos continuamos con el evento programado. Gracias.
Muy buenos días a todos y a todas. Vamos a continuar con las actividades programadas para el día de hoy en nuestro Congreso Internacional sobre Biotecnología, Ingeniería y STEM. Y toca el turno escuchar al doctor Víctor Manuel Ruiz Valdivieso, quien expondrá su conferencia Omic Sciences and Their Application in Sustainable Agriculture. Y previo a eso, pues me permitiré leer una pequeña reseña sobre el doctor. Él y maestro en Ciencias en Ingeniería Bioquímica egresado del Tecnológico Nacional de México, Campus Tuxtla Gutiérrez. Posteriormente, cursó sus estudios de doctorado en Ciencias en la especialidad de Biotecnología en el Centro de Investigación y de Estudios Avanzados del Instituto Politécnico Nacional. Actualmente es profesor investigador del Instituto Tecnológico de Tuxtla Gutiérrez. Es miembro del Sistema Nacional de Investigadores Nivel 2, del Sistema Estatal de Investigadores, miembro honorífico en Chiapas y profesor con perfil deseable PRODEP. Recibió de la Medalla de Oro por Reconocimiento al Mérito Estatal de Investigación en el 2014 por parte del Instituto de Ciencia, Tecnología e Innovación del Estado de Chiapas. Actualmente imparte clases en la licenciatura de Ingeniería Bioquímica, la maestría en Ciencias en Ingeniería Bioquímica y el doctorado en Ciencias de los Alimentos y Biotecnología. Ha dirigido proyectos de investigación financiados por diversos organismos como el CONACYT y el Tecnológico Nacional de México. En cuanto a la formación de recursos humanos, el doctor Ruiz Valdivieso ha dirigido 6 tesis de doctorado en Ciencias de los Alimentos y Biotecnología, 10 tesis de maestría en Ciencias en Ingeniería Bioquímica y 30 tesis de la licenciatura en Ingeniería Bioquímica. Actualmente dirige 2 tesis de doctorado en Ciencias de los Alimentos y Biotecnología, 2 tesis de la maestría en Ciencias en Ingeniería Bioquímica y 5 tesis de la licenciatura en Ingeniería Bioquímica, así como asesora a estudiantes de Ingeniería Bioquímica en residencias profesionales. En materia de productividad científica, ha publicado 110 artículos en revistas indexadas en Scopus y cuenta con un registro de patente y un trámite de registro en proceso de evaluación, así como también ha participado en diversos eventos académicos a nivel nacional e internacional. Pues sin más preámbulos, le cedemos la palabra al doctor Víctor Manuel Ruiz Valdivieso.
Doctor Gerardo, el doctor Víctor tenía un poquito de problemas de intermitencias de conexión, pero ahorita en un momentito ya va a poder estar con nosotros. Esperemos se conecte.
Perfecto, entonces esperemos unos momentos para poder escuchar al doctor.
¿Me escuchan?
Sí.
Sí, muy buenos días. ¿Me escuchan? Voy a compartir la presentación.
Sí, se le escucha, doctor.
Gracias. Ok.
Le solicitamos paciencia al público en general y en un momentito vamos a estabilizar la conexión del doctor invitado.
Hola, ¿me escuchan?
Sí, doctor, ya lo escuchamos.
Sí, lo que no tengo es la opción para compartir, disculpe. Ajá, disculpe.
Me parece ser que la pueden compartir nuestros apoyo técnico, la presentación, Mari.
Y ya, doctor, ya está habilitado como moderador. Usted puede, en teoría debe estar habilitado el botón comparte.
Ok, voy. Gracias.
Lo que pasa es que, bueno, yo no tengo la presentación del doctor. No sé si ustedes la tengan.
Voy.
Sí,
¿Ahí ya,
doctor,
ahí ya lo
ya.
observan? Ok, ya estoy.
Ahí está, ya la estamos viendo, doctor.
Una enorme
Adelante.
disculpa, una enorme disculpa. Ya. Muy buenos días. ¿Sí me escuchan? ¿Puedo iniciar?
Adelante, doctor, lo escuchamos bien
Sí.
y se ve su presentación.
Gracias. Muy buenos días a todos. Eh, distinguidas autoridades, colegas, estudiantes, investigadoras e investigadores que están aquí presentes en, en la organización de este Congreso Internacional. Es para mí un honor y un privilegio estar aquí y poder compartir con ustedes esta conferencia en el marco de tan importante evento académico. Agradezco sinceramente, nuevamente, disculpen las fallas técnicas, a los organizadores por su amable invitación y por brindarme este espacio. El objetivo de esta plática, básicamente, es explorar el impacto de las ciencias ómicas en el desarrollo de una agricultura sostenible y eficiente a través de ejemplos prácticos y resultados científicos, principalmente de cepas microbianas y algunos insumos de interés agrícolas que se han desarrollado aquí en el Instituto Tecnológico de Tuxtla Gutiérrez. En el contexto de la agricultura sustentable, es sabido que vivimos en tiempos complejos donde la agricultura pues es una actividad fundamental para la seguridad alimentaria y la estabilidad de nuestra sociedad, la estabilidad social que perseguimos aquí en México. Sin embargo, se enfrentan grandes retos globales como es el cambio climático, el agotamiento del suelo y la creciente inseguridad por los alimentos, la inseguridad alimentaria. Estos factores están profundamente interconectados y pues nos están poniendo a pruebas, ¿no? A nuestras sociedades. ¿Para qué? Para poder producir alimentos de manera sostenible, suficiente y resiliente. Ante este panorama es necesario o es indispensable replantear el nuevo modelo agrícola convencional y avanzar hacia un paradigma verdaderamente sustentable. Pero, ¿qué significa esto? Significa desarrollar sistemas de producción, básicamente, que no solo aumenten el rendimiento del cultivo, sino que también regeneren los recursos naturales que tenemos aquí en México, aquí en Chiapas, y se reduzca la dependencia de insumos químicos y pues se enfrente a todo lo que es el cambio climático, siempre teniendo una evidencia científica, una evidencia tecnológica. Por ello, es aquí donde existen las necesidades de implementar soluciones biotecnológicas innovadoras. Y es aquí donde la biotecnología juega un papel crucial. Sé que aquí en este foro hay estudiantes, hay investigadores e investigadores, recordando la biotecnología es el uso de los seres vivos y sus derivados o partes de ellos para obtener un bien, un producto, un servicio, aplicando el uso de los sistemas biológicos, llámese un microorganismo, una planta, principalmente. Pero bueno, en la biología moderna se utilizan diferentes áreas del conocimiento que han tenido un gran impulso tecnológico para acercarse a su estudio, como es la microbiología, la genómica, la bioinformática, la proteómica, la bioquímica. entre otras, que nos permiten comprender el potencial que tienen un individuo, un microorganismo y las interacciones que puedan surgir entre ellos, tanto en plantas con los microorganismos. Así surgen las ciencias ómicas. Yo le estoy poniendo aquí en esta diapositiva como un mundo de datos, en el cual el término de ciencias ómicas no es más que un neologismo, que se refiere a una nueva palabra, ¿no? Que nosotros mismos en el mundo de la investigación hemos estado diseñando, implementando. Este involucra un conjunto de disciplinas científicas que estudian de manera integral conjuntos complejos de biomoléculas dentro de un sistema biológico. Válgase, o es importante comentar, biomoléculas pueden ser genes, proteínas, metabolitos, transcritos, entre otros. Así es como actualmente pues tenemos el estudio de la genómica, que estudia el conjunto de todos los genes que se encuentran en un genoma, es el estudio del ADN en su totalidad. La transcriptómica estudia los transcritos, los niveles de expresión que se encuentran en una célula, en una planta, en un microorganismo. La proteómica estudia las proteínas presentes de la misma forma en una célula, en un tejido. Y la metabolómica, por mencionar el análisis de los metabolitos presentes en una célula, en un tejido, ¿no? Esto es importante desde el punto de vista de la agricultura, porque estas herramientas nos permiten identificar, nos permiten hacer diagnósticos de diferentes, en el caso de nosotros, cepas microbianas, para promover el crecimiento vegetal, por ejemplo, fijar nitrógeno, hasta degradadores de contaminantes. Y desde el punto de vista bioquímico, por ejemplo, no solamente estudiar la presencia de algunos microorganismos, sino también la función que puedan tener, ¿no? Ahora, ¿cuál es la importancia, como habíamos comentado? Es la caracterización funcional de genes y rutas metabólicas, que ha sido un reto que hemos tenido aquí en el tecnológico y en el grupo de trabajo, ¿no? Llámese también para su uso en agricultura, y el desarrollo de bioinsumos agrícolas. Entonces, es muy importante las ciencias ómicas en la biotecnología, en la agricultura sustentable. Por mencionar la metabolómica, en el estudio del flujo metabólico, nos ha permitido estudiar los metabolitos que están presentes o que son claves en un cultivo o en la producción de un bioproducto, por ejemplo, un biofertilizante o la detección de algunos agentes antimicrobianos, antifúngicos, antibacterianos, por ejemplo. El rastreo de las rutas, ¿para qué? Para poder diseñar o optimizar procesos y poder inclusive aplicar estas técnicas biotecnológicas para producirlos a nivel de matraz o a nivel de bioreactor. Pero bueno, vamos a hablar un poquito acerca de lo que tenemos aquí en la carrera de Ingeniería Bioquímica y en particular en el área de biotecnología vegetal y microbiana del Tecnológico de Tuxtla Gutiérrez, ¿sí? Un caso de estudio ha sido aquí en el grupo de trabajo el proyecto del volcán Chichón, así le he estado denominando. Es un proyecto que iniciamos en el año 2015 con un proyecto que fue aprobado por el extinto CONACYT, que se titula El análisis metagenómico de la estructura de las comunidades microbianas y su expresión genética en el lago cráter del volcán Chichón. Esto fue en la modalidad de joven investigador. Ustedes aquí en esta imagen observan este sitio hipertermófilo, así le llamamos, el cual se encuentra ubicado de acuerdo a este mapa principalmente en el norte del estado de Chiapas, ¿no? Este volcán tiene en su interior un lago cratérico. Es un lago cratérico que tiene ciertas características que voy a mencionar posteriormente. Por mencionar, es el único volcán que en este momento en México tiene un lago con estas condiciones, ¿no? Como se menciona aquí, el Popocatépetl en el 94 hizo la erupción y desaparece ese lago, ¿no? Ese lago de agua caliente. Si hay fumarolas en el Popocatépetl, en el Nevado de Toluca, más sin embargo, en el Chichón se encuentran estos hervideros, este lago como tal, ¿no? De agua ácido térmico. Aquí observamos algunas fotografías de la zona del volcán, en la cual vemos desde un punto de vista panorámica cómo se observa el lago cráter, luego hacemos acercamientos, vemos las fumarolas y algunos sedimentos. Aquí a ver si se puede reproducir este video, no sé si escuche. Esa es la parte panorámica. Esta foto fue de hace 5 o 6 años. Hemos hecho ya 11, 12 muestreos en este ambiente extremo. Estos son algunos de los hervideros. Es sorprendente poder estudiar, llegar, analizar, colectar en este sistema, ¿no? Pero bueno, ¿cuál es lo importante y por qué quiero mencionar esto para el uso en la agricultura? Nosotros en las primeras etapas, aquí vemos una serie de artículos ya publicados. Todo esto tiene un sustento del grupo de trabajo, en la cual evaluamos altas temperaturas desde 50 a 96 grados centígrados. Tiene también alto contenido de metales pesados y ambientes ácidos, ¿no? Donde el pH puede estar entre 1.9 hasta 6, o inclusive se mantiene en 1.9 a 3, ¿no? Por lo regular. Entonces son ambientes hiperextremos que tiene muchas condiciones, ¿no? Que lo hacen muy importante. Entonces, de eso nacen la pregunta, ¿no? Si había vida en el lago cratérico. Desde su inicio, los estudios estaban más relacionados a estudios de vulcanología, a estudios geoquímicos, a estudios más del análisis físico-químico del agua y sedimento y las fumarolas, ¿no? Y la actividad vulcanológica que pueda tener aquí este ambiente, ¿no? Pero entonces nosotros empezamos con esta parte de cultivar en el laboratorio, generar medios de cultivo, poder aislarlo, y lo veíamos como un ambiente, o lo vemos, ¿no? Como un ambiente natural muy importante que tenemos aquí en el estado de Chiapas. Aquí es parte de la ubicación. Hay que irnos de aquí del Tecnológico de Tuxtla, 4 o 5 horas, caminar 2 o 3 horas, descender con todos los protocolos de seguridad para poder tomar las muestras, dependiendo del objetivo clave, y poder estudiar estos microorganismos, ¿no? Así es como hicimos los primeros estudios de diversidad y estructura de comunidades de bacterias extremófilas en sedimentos del volcán El Chichón, con ahora la doctora Clara Ivette Rincón Molina, con quien publicamos el primer artículo en la revista Geomicrobiology Journal, en el cual evaluamos la diversidad de la estructura en 2 temperaturas y empezamos a hacer los primeros estudios metagenómicos con base en el gen 16S, rRNA. Entonces hicimos la detección de estos microorganismos y en las primeras etapas, esta gráfica me gusta mucho. De hecho, este muestreo ayer le comentaba a la doctora Claribe, me recuerda bastante cuando empezamos con los primeros muestreos, con lo poco o mucho que teníamos aquí en el tecnológico, aquí en el sureste mexicano, a medir esos parámetros de pH, de temperatura, y detectamos la presencia de algunos géneros bacterianos principalmente, como Metylobacterium sulfobacillus, de sulfurispora, Painibacillus, que después les voy a platicar la historia de esto, Bradyrhizobium, entre otros, ¿no? Para quienes estamos en el tema de ecología microbiana y la agricultura, es sabido que principalmente Painibacillus y Bradyrhizobium son de interés agrícola. Entonces nos empezó a llamar la atención cómo en este estudio metagenómico, donde extraemos el ADN total, construimos las bibliotecas metagenómicas. Estamos hablando que esto lo hicimos 2017, 18, y aquí pues la publicación ya 2019. era cuando estaba un boom, ¿no? Hace 8 o 9 años del Illumina MySeq. Y entonces empezamos a hacer estos análisis y estos descubrimientos. Más sin embargo, no habíamos cultivado, ¿no? Los microorganismos. Por eso se los voy a platicar. Y fue como nace la idea de poder cultivar, estudiar diferentes grupos ya posteriormente de comunidades o microorganismos extremófilos aislados del volcán. Pero bueno, a través de otras herramientas bioinformáticas que actualmente juegan un papel muy importante, igual en el año 2016, 17, 18, empezamos a implementar estas técnicas de predicción metabólica empleando el gen 16S. Este es un análisis, lo pongo tal y como empezamos a hacerlo en estos estudios, en el cual hicimos el acercamiento en aquel momento usando PyCrocs, ¿no? Y ahorita existen otros software, otros programas para poder hacerlo. Y bien, detectamos principalmente metabolismo de carbohidratos, metabolismo de aminoácidos en mayor abundancia, así como también los procesos de replicación y reparación celular, ¿no? Entonces nos empezó a dar, así como compuestos xenobióticos, en alguna de estas partes se menciona, aquí en esta parte, y pues empezamos, ¿no? A hacer los primeros acercamientos in silico. Actualmente, y mis compañeros ponentes, investigadores que están acá, esto ha cobrado un boom, el análisis in silico, el análisis computacional. Entonces, en aquel momento nosotros siempre teníamos la duda si realmente esto iba a ser evidente. Sí, estoy hablando de hace 7, 8 años. Y entonces empezamos a hacer ya los estudios bioquímicos para poder estudiar y evidenciar también lo que nosotros estábamos encontrando, ¿no? Así fue como la ahora también la doctora Bepsianay Peña Ocaña, quien trabaja en el Departamento de Bioquímica en el Instituto Nacional de Cardiología, en el equipo del doctor Ricardo Jasso Chávez. Hicimos los primeros estudios del metagenoma completo en 2 tipos de muestra, le voy a llamar así, que es la muestra de agua y muestras de sedimento del lago cráter del volcán El Chichón. Aquí, pues fue de mucha importancia el estudio o para entender cómo estaban en el lago cratérico o en este ecosistema, en estas condiciones únicas, y poder entender el entorno, ¿no? Y no solamente poder estudiar la presencia de las comunidades, con el metagenoma podemos detectar al mismo tiempo la presencia de bacterias, hongos, arqueas, y también detectamos la presencia de virus, ¿no? Recuerdo con Bepsi platicábamos ahí acerca de cómo detectamos la presencia empleando, como dice ahí, un enfoque multiómico, en el cual empleamos la metagenómica, empleamos transcriptómica, la bioinformática, no solamente para entender quiénes están presentes, qué microorganismos están presentes, sino qué funciones puedan estar realizando y para poder evaluar su futuro potencial biotecnológico. Entonces, estas ciencias ómicas nos llevaron al futuro, ¿no? Para poder tener una aplicación, un uso, ¿no? Y así es como muchas especies que fueron en ese momento detectadas, pues tienen potencial en la remoción de metales pesados, tienen potencial ahora en agricultura, en el metabolismo del azufre, en el metabolismo del arsénico, en diferentes áreas, ¿no? En la bioremediación, en la minería, ¿no? Entonces, y también en la síntesis industrial. Entonces, este tipo de investigaciones no solamente es generar conocimiento, sino que también nos va contribuyendo o generando un panorama acerca de cómo es una herramienta esencial para aprovechar el potencial oculto que tienen este tipo de microorganismos, ¿no? Aquí por mencionar Hydrogenobacterium, entre otras arqueas, entre otras bacterias fueron identificadas. Nuevamente identificamos Panibacillus, Bradyrhizobium. Pudimos también a través del shotgun, ahorita no lo menciono, pero aquí está el doctor Luis, en esta foto el doctor Luis Eduardo Servín, con su equipo, logramos identificar una nueva arquea, la primera arquea identificada en este ecosistema tan importante, ¿no? Pero bueno, la historia continúa. Aquí se hicieron algunos estudios, por mencionar Bepsi, o con la doctora Bepsi, pudimos coincidir y vincularnos con algunos grupos como el Centro de Astrobiología, que también está asociado a la NASA, allá en España, principalmente con la doctora Elena González Toril y el doctor Javier Tamames, ¿no? Entonces, se hicieron algunos estudios, inclusive también generamos medios de cultivo, por eso dice cultivos Langley, que está en proceso de patentamiento, por cierto, para poder cachar, para poder evaluar, estudiar bioquímicamente y producir metabolitos de interés, ¿no? Entonces, en este preámbulo, tanto el análisis de secuenciación masiva por 16S, que se hizo con la doctora Claribet, después con la doctora Bepsi, ¿no? Con los metagenomas, pues nos dio una idea del potencial que pueda tener y muchas de las bacterias pues inclinaban hacia el uso de la agricultura. Así es como el día de hoy la estudiante de doctorado Martínez López, la maestra Nancy Abril, pues estaba hablando del potencial de biocontrol y promoción del crecimiento vegetal de la cepa BCA1 del Panibacillus en diferentes tipos de cultivos, ¿no? Ella ha logrado ya aislar, hemos diseñado medios de cultivo y se han logrado evaluar el potencial de las cepas principalmente de Panibacillus. Como el agente de biocontrol, se han identificado genes, se secuenció el primer genoma de una bacteria de aislado, ya está anunciado en esta revista tan importante de microbiología, que es el genoma del Panibacillus, y también se han identificado genes, rutas metabólicas claves asociadas a la producción de compuestos antifúngicos, de compuestos bioactivos y algunos biosurfactantes, algunas fitohormonas, por ejemplo, como el ácido indolacético principalmente, ¿no? Entonces, estos descubrimientos que actualmente han estado realizándose con la tesis doctoral de Abril, que ya próximamente también está a titularse, pues nos dan una alternativa sostenible a los agroquímicos convencionales, ¿no? Y ella detectó por técnicas de cromatografía la presencia de algunos metabolitos de interés como la iturina, por mencionar, que tiene muchas aplicaciones prometedoras, le llamemos así, para su uso como biofertilizante y principalmente para la protección vegetal en cultivos agrícolas. Ahora, Abril, pues también hace el estudio de los genomas, algunas pruebas que se han hecho también a nivel invernadero. Estos proyectos pues también tienen un enfoque social, tenemos que ir con los productores de tomate, de chile, de aquí, de la región de Chiapas. Yo he hecho hoy a la 1:30 un estudiante que tenemos en común con la doctora, con la maestra Nancy. Jenny se va a titular, con ella hemos aislado algunos fitopatógenos de cultivos de mango, también para poder estudiar el efecto, hacer las pruebas in vitro y también las pruebas a nivel de campo, ¿no? Aquí, por mencionar, son los estudios transcriptómicos, metabolómicos que hemos hecho. Se han descubierto la producción de sideróforos, algunas enzimas, antifúngicos como la iturina y algunas hormonas.
Me parece que el doctor está experimentando otra vez problemas técnicos, así que seamos pacientes y esperemos que pueda conectarse pronto nuevamente. Les pido un poco de paciencia. El doctor está trabajando en resolver el problema técnico. Esperemos que pronto pueda conectarse. Vamos a dar tiempo para que el doctor se logre conectar. Y mientras, si les parece, vamos a dar entrada a la conferencia que está programada a continuación, para aprovechar el tiempo. Toca el turno de la presentación del doctor Federico Antonio Gutiérrez Micelli con la conferencia Experiences in Seed Preactivation by Physical Method. Y para dar una reseña del doctor, él es ingeniero bioquímico en productos naturales, con maestría en ciencias en biotecnología y el doctorado en ciencias con especialidad en biotecnología. Pertenece al Sistema Nacional de Investigadores, nivel emérito, del Sistema Estatal de Investigadores en Chiapas y profesor con perfil deseable PRODEP. Actualmente imparte clases en la licenciatura de Ingeniería Bioquímica, en la maestría en Ciencias en Ingeniería Bioquímica y en el doctorado en Ciencias en Alimentos y Biotecnología. Ha graduado 12 doctores en ciencias, 32 maestros en ciencias y 52 ingenieros bioquímicos. De acuerdo a Scopus, tiene 148 artículos publicados, 2013 citas y un índice H de 22. Su tema de investigación es el metabolismo primario y secundario de vegetales cultivados con insumos orgánicos. Entonces, si le parece, doctor Gutiérrez, tiene la palabra.
Gracias, muchas gracias. Ya estamos aquí. Ahorita voy a poner la presentación. A ver si
Les recuerdo que pueden colocar sus preguntas en el chat y al final de la conferencia, pues le daremos lectura.
Doctor Micelli, igual estamos aquí para apoyarlo en soporte técnico si usted está teniendo alguna complicación para proyectar su presentación.
¿ahí me escuchan?
Sí, doctor.
Sí. Muy bien. Ahorita estamos, no carga la presentación. pero en un momento lo hacemos. A ver si quieres. Es abrir, ¿no? En PowerPoint y directamente lo carga. Abrir.
Doctor Ruiz, no sé si me escucha, no sé si sería posible que usted proyectara su presentación y continuara donde se quedó en lo que resuelve su problema técnico el, el doctor Gutiérrez. ¿Me escucha, doctor Ruiz? Creo que seguimos teniendo problemas técnicos, así que nos queda solo esperar. Seamos pacientes a que logren resolverlo.
Como que no es.
Perdón, aquí aparece un anuncio que dice que no se puede mostrar el contenido, no se puede descargar el archivo en la plataforma. No sé si podrían ayudarnos.
A ver si te veo, Miss, y le digo, ¿por qué le estás trabajando en su comité de vuelta?
No me oyeron, yo creo.
Sí, doctor,
Ajá.
¿qué le parece? Nosotros estamos de soporte técnico. ¿Por qué no nos
Sí.
hace llegar su presentación por correo electrónico? Nosotros lo proyectamos y ya usted nada más imparte la charla.
Perfecto, ahorita se lo mandamos.
Perfecto.
Gracias.
Y doctor Víctor Manuel Ruiz, no sé si me escucha, para que en todo caso pudiéramos mientras continuar con usted. Me parece que no, entonces vamos a esperar que el doctor Gutiérrez mande su presentación para poder comenzar con su charla.
Hola, buenas.
Sí,
¿Sí
doctor
me-
Ruiz. Sí.
Una disculpa, no me di cuenta que ya estoy viendo el video. De hecho, me reportó un colaborador. Pensé que estaba conectado. Una enorme disculpa a todos.
No
Igual el
sé,
doctor tiene complicaciones. Casi nunca se
Sí.
nos va el internet en el instituto.
Y justo hoy, ¿verdad? A veces pasa.
Sí, parece que lo viéramos.
Ley de Murphy.
Ajá, y él está allá en su oficina, que somos colaboradores aquí cercanos, pero no.
Pues no sé si quiera, mientras usted